Welcome all. In today's lecture, I will talk about UniProt, which is one of our database of protein sequence and function information. It is a huge database that gives a very detailed information about any protein. So in this session, I will just start by giving a brief introduction of UniProt, what UniProt is, and then I will take an example of one of the protein that is involved in immune system, and we will use that protein to see what type of information we can get from UniProt. So let's start. UniProt is like the abbreviation. It stands for our Universal Protein Resource. It is a comprehensive resource for providing information about protein sequences and functions. This database is further subdivided into sub-databases, like we have a UniProt knowledge base, which is again divided into sub-database. That is one of that sub-database is known as SwissProt, and another one is Tremble. The other sub-database is UniProt reference cluster, also known as UniRef. And the third one is UniProt archive, which is UniPark, also known as UniPark. And then we have Proteome. I will discuss in a moment what these different sub-databases are. Start the hands-on session. So the web link for UniProt is uniprot.org, and this is the home page. And you can see that we have different sections or sub-databases. Like we have UniProt KB, UniRef, UniPark, and Proteomes. We will use this UniProt KB, and I will discuss UniRef and other databases in other lectures. So UniProt KB, I mean, you can see that UniProt knowledge base is having two sections. So one is SwissProt and the other one is Tremble. Now, the SwissProt is that section in which all the information they are manually annotated and reviewed. So scientists review that information and then they become a part of SwissProt. However, in Tremble, which is translated European Molecular Biology Laboratory section, they have the information in this section. They are automatically annotated and they are not reviewed by a scientist. So we can use, we are going to use this UniProt knowledge base by giving our protein of interest, and we'll see that what type of information we can get. In addition to that, there are certain tools you can also access from this homepage, like we have BLAST and sequence alignment, and another additional feature as well, like literature citations, taxonomy, information about diseases, subcellular locations, and cross-reference databases. So I will discuss these different features in some other lectures. But for now on, I will discuss, I will use one of the protein that is involved in immune system and see that what type of information we can get by using UniProt database. So one thing that here I'm going to get the information from UniProt KB. So if you can click on this arrow, you will get all those sub-databases. We are interested in UniProt KB, so I'm just going to cross that because UniProt KB is already selected. So I will write a protein name. This is granzyme B, which is one of protease, a serine protease that is involved in immune system. So I want to know that what type of information I can get about granzyme B. So then I will search the information. So here you can see that we have a list of entries and a different information. One important thing is that on left hand side we have filters. So if you click on that, a search result page is subdivided into filter panel on the left and the actual result table on the right, occupying the majority of the screen space. So if you want to, the bluish are the color tags. They are all that will contain all those information that belongs to SwissProt. So these informations have been manually annotated while the blue tag, the information having blue tag, they are and not annotated manually. They they are taken from Tremble. You can see that we have a total number of entries as 1292. So we have to refine our search. If I just click on that just to make it more clear, how do we refine our search? Now I'm interested in knowing the information about granzyme B, but I'm more interested in human granzyme B. So how I will refine my search? If we use this advanced feature and we just want to add or refine our search by this way. So here you can see that we have and, are, and not. So these three, they are known as Boolean operators, and these Boolean operators are always written in caps. And with that, you can refine your search. For instance, I want to refine my search by using organism. So if I click on that all and select organism, and here I write human and then search again, you can see that the search now we have 47 entries. So initially when I selected when I wrote granzyme B and search, I got 1200, more than 1200 entries, but in this when I refined my search, I got only 47 entries. So on the left hand side we have a panel which you can use for filtering your information, and here in this section we have a sort of information in a column form. So you can select different entries, you can download them in different formats. I will discuss this one later. You can, if you just click on column, you can add or you can remove these columns to make your search more clear, and whatever information you want, you are interested in, you can keep only that column. For instance, if you want to remove this column, you can remove that, and you can just keep those columns of which you are interested in. So for the time being, I'm just going to unselect that. I am just going to select only the top one, which is the entry name is P10144, and let's see what we will get. So this is the detailed information we can get for granzyme B, and P10144 is the UniProt KB entry name, and if you scroll down, you will have a list of information available for granzyme B. Now on the left hand side, you can see that we have these different sort of options. If you want to, if you're not interested in going through all those details, if you are interested only in expression, you just click on that expression section on the left hand side. You will get only the information about expression. If you are interested in the structural information, if you just click on that, it will guide you to that particular section. So let's start from the beginning. Okay. Now this is the this is the display section. So we have the entry, whatever the entry name is, the protein name is granzyme B, gene is granzyme B, organism is Homo sapien, and status is reviewed. So as I mentioned before, that if we have that brownish or yellowish tag, that means that it has been reviewed manually, and it is a part of Swissprot. Okay, this is very important annotation score. So in this case, it is 5 out of 5. If you just click on that, the annotation score provides a heuristic measure of the annotation content of UniProt entry or proteome. So it doesn't give the information about the accuracy, but the level or amount of information, simple word, that whatever information we have about that particular entry. So the information is sufficient, information is good. It is the annotation score is 5 out of 5. In this site, going back to the left hand side, you can just click on the publication. It will give you all the list of publications that the information is taken from, and that becomes a part of this whole for this particular entry. So we have feature tables. If you are interested in in a tabular form, you can just click on that and it will give you a sort of information in a tabular form, in a tabular view or in a tabular form. Let's start the information what we can get for granzyme B by using UniProt KB. Now this section will give you an information about the function. It's just a summary that what type of function granzyme B performs, where it is it is involved in which particular function. To support that information, there is again a sort of a sticky note that if you click on that, you will get the publication list that support this information. So we have this section is catalytic activity, and that gives you an information that as this granzyme B is a protease, so it is going to the catalytic activity section gives you an information that which is a preferable cleavage site. So in this case, we can see that the granzyme B is going to cleave after aspartic acid. So so that's the preferable cleavage site. So that is also known as aspase, because aspartic acid is acidic amino acid. So any protease that cleaves after aspartic acid, they are known as aspases simply. There are also reports that they show cleavage, like after methionine and serine and asparagine. But the preferential cleavage site is right after aspartic acid. If you want to know more about these cleavage sites and from where what is the source of this information, you can just click on that publication. It will give you all the list of publications from which this information is taken. This is the enzyme code. As you know that every enzyme, the name of enzyme, when we talk about the enzyme nomenclature, every enzyme is given a particular code, and that code gives you an idea that to which main class and which particular reaction enzyme is going to perform. So this 4-digit code, which is 3.4.21.71, is a code given for granzyme B, to granzyme B. Then this section is activity regulation. Whether it is a protease. So is there any inhibitor, or is there any regulator that stops the activity of granzyme B? So that information is also provided, that this is the inhibitor, and you can check further details on the by clicking this publication and then go to a particular publication and have a look of it. As this is a serine protease, so it will have an active site. An active site is that region where a particular reaction takes place. In this granzyme B, there are 3 amino acids which are very important, and they become they are very crucial in the active site. So if we mutate these 3 amino acids in the active site, then the enzymes become inactive. So one of the amino acid that is involved that plays a very important role in the catalytic activity, that is present available in the active site, is histidine 57, and the second is aspartate 102, and third one is serine 195. So these 3 amino acids, they become a catalytic triad, like we have in a serine proteases. Granzyme B is one of a serine protease. So granzyme B also has these 3 amino acids in the active site, and these 3 amino acids in the active site are very much crucial for performing the the enzyme activity of granzyme B. Now we have, if you click on this number 1 active site, it will take you to a region that number 1 amino acid that is a part of the active site is located at position 64. If you click on this 64, it will take you to a site where you can see that this is the sequence. So you can see that we have histidine, which is highlighted. So this is one amino acid that is located in the active site, which is histidine. In chymotrypsin, we represent them as histidine 57 with reference to chymotrypsin amino acid numbering. But generally, it is, it can be named because the position in different amino acids, different proteases, they vary. Similarly, we have aspartic acid at 102. If you click on that, it will take you again to the page where you can see particular amino acid, and then 203, if you click on that, it will take you to a region that is serine 195 in chymotrypsin, but in this case we have serine 203. So the other one, the fourth one, is amino acid that is located at position 228 in granzyme B. So this amino acid is very much important because that basically defines that what particular specificity a serine protease will have, particularly in this granzyme B. So granzyme B is an S-phase, meaning that it is going to cleave after aspartic acid. But why it is going to cleave after aspartic acid is because of the presence of amino acid at position 228. If you click on that, it will take you to that particular amino acid. So this is the graphical view, and you can see these different amino acids at different positions. So this is 64, that one is 108, and this one is 203, and this is 228. Then we have another section which is Geomolecular Function. If you just click on that, it will give you the information, like the information is taken from Gene Ontology Database. That gives you an information about the function, about the biological processes, that in which particular processes the granzyme B is involved. And in addition, it will also give an information about the subcellular location of a granzyme B inside the cell. So this section will give you the information about the oral function. Geobiological processes will give you information about the particular processes in which this enzyme is involved, like we have apoptotic process, cytolysis. So these are different processes in which granzyme B is involved. Then going back, I'm not going to cover all those details because otherwise it will be a bit long session. So this section is about names and taxonomy, and you can see the summarized information, like what is the recommended name, enzyme numbers, alternative names, gene name, so organism is Homo sapiens, taxonomic identifier. If you just click on that, it will take you to a taxonomy page. And then proteomes, if you remember, there is one database in UniProt that is proteome. So this is the proteome database ID. If you just click on that, it will take you to that sub-database, and you can check the information, the relevant information. So this is the organism specific database which you can access. Now this one is the subcellular location, and here you can see that we have this portion, this lysosome, and you can see that if we have this bluish color, it means that it is automatically annotated, and this yellowish color means manual annotation. So you can see that we have these granules, and these granules means that granzyme B is located in a lysosome. In addition, it is also present in the secreted form as well. Then we have another section which is Pathology and Biotech section, and you can see that if there is any disease related to granzyme B, whether or it is reported in any disease, so this information can be seen from different databases, because the link is there. So if you are more interested, you just click on that link, and then it will guide you to that particular database. For example, if we have DIS GENet, if you just click on that, it will take you to a database that will give you any information, relevant information, or the involvement of granzyme B, or the role of granzyme B in any disease. So it will give you that information. Similarly, we have for example, the chemistry database. If you just click on that Campbell, it is a database of bioactive drug-like small molecules. Then we have guide to pharmacology. So genetic variation database, we have Biomuta, we have DMDM. So if you are interested in information about the pathology and biotech, by the way, if you just click on that, it will give you a brief introduction that what type of information you can get from this section. So this section provides information on the disease and phenotype associated with that protein. And you can access these different databases to to know about the granzyme B, its involvement in pathology, or diseases, or its phenotype. So then we have the other section, which is PTM, or processing, which is if you just click on that, it gives you information about if there is any post-translational modifications or any processing event. So this is an enzyme. So generally all enzymes are proteases. They are secreted, or they are synthesized in their inactive form, which is pro form. In addition to that, they do have certain other sequences, certain other peptide sequences used are involved in different functions. For instance, if you take into consideration this granzyme B, we have the total length of granzyme B is 247 amino acid. However, this 247, the whole sequence is not a mature sequence. So we have a signal peptide that the subsection of denote the presence of N-terminal signal peptide. So signal peptide is essential to direct initially synthesized protein into endoplasmic reticulum. So from amino acid number 1, starting from N-terminus to amino acid 18, there will be a signal peptide, and the relevant information is can be seen by looking into the peptide. So this is the graphical view. And then we have a propeptide. So propeptide, as I mentioned before, that all serine proteases, they are synthesized, all proteases rather, they are synthesized in the form of a propeptide. So propeptide is the inactive form of protein, and usually propeptide is located at the N-terminus. So if we want to make a protein active, we have to remove this propeptide. In granzyme B, the propeptide is just 2 amino acids. So and then the numbering of these amino acids is like number 19 and number 20. If you just click on that, it will take you to that particular position. For instance, I will show you here. So you can see that we have GE, these 2 amino acids, they are the propeptide, and graphically it is shown here. And then the main chain, or a mature protein, it starts from 21 to 247. So this is like a complete the sequence information of granzyme B, and that says that the total length of granzyme B is like 247 amino acid, and in that 247 amino acid, the first N-terminus 18 amino acid, they are used, they work as a signal peptide, then followed by 2 amino acid which are a propeptide, and then the last 227 amino acid constitute the mature protein. Similarly, the amino acid modifications, if we have any modification, like we have post-translation modification, there are a number of post-translation modifications, like we have disulfide bridges between cysteine amino acid, glycosylation. That information can also be, we can get this information from here as well. For instance, the disulfide bridge, is located between amino acid number 49, so cysteine 49 and cysteine 65. There will be a disulfide bridge. Then the amino acid at position 71, that will be glycosylated. Amino acid at position 104 will also be glycosylated. And again, we have these the amino acid are cysteine at position 142 and 209. They will make a pair. They will make a disulfide bridge, and then another pair, which is amino acid at position 173 and amino acid at position 188. So these both amino acid, they are cysteine. However, the cysteine located at position 173 and at position 188, they will make a disulfide bridge. So we can see that there are three disulfide bridges, overall three disulfide bridges in granzyme B, and there are two amino acid that are glycosylated. So you can see that we have this information available that gives you an idea, that gives you the information that what particular amino acid is involved in what different type of post-translation modification. So some other relevant information you can see from here. And then the expression. So expression provides information on the expression of a gene at the mRNA or protein level in a cell or a tissue of multicellular organism. So we have here you can see that they have used staphylococcal enterotoxin, which is enterotoxin and A, that is used to observe the expression of granzyme B by using peripheral blood leukocytes like white blood cells, and the information about this expression by these white blood cells by using enterotoxin A as an inducer. So whether they are expressed in granular sites and what type of expression is that, so that information you can get from this section. Then this section will give you the interaction section. It will give you the information that whether in a cellular environment granzyme B is going to interact with other proteins or not. So we can see that in this case granzyme B is going to interact with two proteins. So one of the protein is SRGN, which is sarglycine protein, and another one is perforin, which is a pore-forming protein, and it facilitates the entry of granzyme B into a target cell to initiate apoptosis. The details can be seen here. About perforin and sarglycine, which is SRGN. If you're interested to know more about these interaction, you can go to this. Just click on that link, and it will take you to that page, and you can see the interaction. So in addition, there are other databases like BioGrid, Intact, and Mint and String. They also provide the information that whether what other proteins granzyme B is going to interact. So if you just click on that, it will take you to that page and give you a detailed information about the interaction. So we have another database, which is a chemistry database, and binding DB. This is the if you search binding DB database by using this entry, it will give you a sort of information that what small molecules are available that can inhibit the activity of granzyme B. What different molecules can be used as an inhibitor of granzyme B. Similarly, okay, now this section is very important. This is every section is definitely important, but this section gives information about the structure of granzyme B. So you can see that we have two structures. The method through which these structures are solved, that is X-ray. The resolution is given, chain information is given, position is given, and the links which you can use to further explore the structural features. For instance, you can go to PDB Europe section, and you can go to the PDB page, which is RCSB. Similarly, you can go to PDB Japan and PDB Sum. So these different databases will give you different information about the structure. So you can go and check these website if you are interested in studying the structure in more detail. You can also play around with the structure in this small section by using this bulletin viewer PDB structure viewer program. You can also play around by using these different options. For instance, you can expand that, you can change the background, you can further play around with the structure. You can also just click on that and you can see the amino acid information at the top. So that portion gives you all about the structural information of granzyme B. If there is no structure available, so we will not have about any protein, we will not have this section. Below we have a secondary structure information, and the legend you can see that blue represents helix, and similarly like the gray represent PDB structure node for this area, and this the other colors show different features which you can see from here. Then we have family and domains. So if you just click on that, it will give you that to which particular family or a domain in a protein it belongs. That shows that the domain is starts from 21 to 245, which is the sequence of mature granzyme B, and that is like known as peptidase S1 domain. If you just click on this sticky tape, it will give you that information. And the total length is 225. So basically granzyme B belongs to peptidase S1 domain. It is, I mean, a protein family based on the characteristic secondary structure pattern in a protein. Then we have some other sequence related features and phylogenomic databases, family and domain databases. You can search each one of them and can get an idea. We have sequence information. For instance, this is the amino acid sequence. If you want to download that, you can just click on FASTA and it will be downloaded. And then we have computationally mapped potential isoform sequences. So if you're interested in the isoform of a particular protein, then you can check that whatever available information is also shown here in this section. And then going down to the other section, like for example, if we have this the experimental info, so if there is any conflict about a position of amino acid, you can also see that, like sequence conflict at position 32 and 33. Similarly, there is a conflict at position 72 and 212. So you can see that if there is any conflicting reports about sequences, it is also shown here. These are, we have 3 natural variants of granzyme B, and these variants are like at position 55, position 94 and 247. So in which R is changed to Q, and similarly proline to alanine. So similarly, you can see the other one as well. So these are natural variants. And then the next section. Okay, by the way, if you are more interested in that, we have these. The links are available here in this section. You can just click on that and go to that particular page. We have this section that gives you the information about similar proteins, and those proteins that are, we have two different sections for that. So granzyme B, the other proteins that are 90% similar to granzyme B, we have those the proteins listed here, they're 90% similar to granzyme B. However, if you are more interested with those sequences that are 50% showing 50% identity, you can just click on that and you will have a list of other entries as well. By the way, this information is located in one of the sub-database which I mentioned in the beginning, that was UniRef. So all the information or entries, they are stored in UniRef based on the sequence identity. So we have a 3 list of clusters One cluster is that, I mean, all those sequences that are 100% similar, they are placed in one cluster, and then we have another cluster which is 90% similar, and then we have the last cluster. So all those sequences that are 50% similar are then placed in another cluster. So basically this information is taken from UniRef, which is another sub-database of UniProt. Then we have cross references, all those literatures available. If you want to read them, you can just go and check. So we have these different information available. It's just like a summary of what we have done earlier. So yeah, this is the entry information. We have entry name and accession, entry history, 1999. I mean, it was the first time when it entered into this database, and last modified date is June 2021. And that's how we get the information. So you can see, we have a complete list of all those information on that side. But if you are interested in a particular information, you just click on that sections on the left hand panel, and it will take you to that particular region you are interested in. For instance, you are interested in sequence information, you just click on that and it will take you to the sequence page. So that's how we use UniProt to get the information about any gene of interest, and you can see that what type of information we get by using UniProt database. So that's all. See you in a next lecture and next topic. Thank you very much.